Vamos a dar inicio a la charla magistral número 6. Va a estar desarrollada por la doctora Stephannie Kaweckie y el título es Prevalencia de bacterias resistentes a antibióticos en aguas de la cuenca baja del arroyo de Ballona. Stephannie Kaweckie es asociada al Laboratorio de Biología de la Universidad de Loyola en Marymount, tiene una maestría en Ciencias Ambientales y su especialidad es la biología de microbacterias. Le damos la bienvenida y le pedimos que pase a dar su presentación.
Knowing myself, I would trip on that, so I'm just trying to push it back a bit. Hi, again, my name is Stephannie Kaweckie, and the work that I'm going to present today is the research that I did to complete my master's thesis. The title is The Prevalence of Antibiotic-Resistant Bacteria, and also known as ARB, in waters of the Lower Ballona Creek Watershed, Los Angeles County, California. You might recognize this name right here, and that's because he is coming up right after me, Dr. John Dorsey. So you will be seeing that a lot of topics that I touch on will actually overlap with John's presentation. So maybe you can play a little game and figure out how many similarities both of our presentations have. So first I'd like to give an introduction to antibiotics, which are compounds produced by bacteria and fungi, which are capable of killing or inhibiting competing microbial species. So why would microorganisms do this? Well, it's a tough world out there. So if you have this ability to create antibiotics, you can use it as a form of chemical warfare against competing microbial species, therefore doing better to obtain your resources in your environment. So in 1928, this was discovered by Alexander Fleming. This was during a time when many people were actually dying due to bacterial infections. There was no cure for illnesses like this. So Alexander Fleming decided that he would study bacteria in an effort to make a discovery to find a cure for these illnesses. So he used petri dishes to grow his bacteria. What a petri dish contains a jelly-like material that has lots of nutrients, so the bacteria will grow well in this environment. So it's a way to visualize and work with bacteria. What ended up happening though during his experiments is before the weekend was about to happen, he got a little sloppy and he actually left his petri dish open. And the issue with that is that microorganisms are able to travel through the air. So if you're working with bacteria and working with a nutrient-rich agar, you have risk for contamination. So that's exactly what happened. He came in after the weekend to his lab and he saw that there was a contamination on his petri dish. So over here, he inoculated his bacteria as normal. When he first saw that, he probably assumed that he was going to have to throw his sample away and everything was ruined. But before doing that, he actually noticed that there was this zone of inhibition. So that led him to conclude that this colony, fungal colony called penicillium, was actually producing some compound that wasn't allowing bacteria to grow in its surroundings. So this was a significant milestone in public health, and it led to the use of antibiotics in humans to cure bacterial infections. And since then, excuse me, I jumped ahead a slide. Over here, we can actually see the benefit that antibiotics have brought to human health. So over here is a figure of infectious death rates in the US between 1900 and 1996. So infectious means viral and bacterial, so this figure encompasses both aspects, which is why we see there was a peak in when there was an influenza pandemic. But the main point of this figure is that we see with the first introduction of penicillin around the 1940s, we have a major drop in infectious death rates. So again, just showing wow, what a great contribution this was to public health. So since then, more antibiotics have been discovered and also synthetically created. And what was also discovered is different antibiotics work differently according to whatever bacterial species you're infected with. So over here, we have the top prescribed antibiotics in 2013. So some of you may be familiar with these antibiotics. Maybe you have taken it at some point in your life. And you know, they're a great thing. They work and they get rid of bacterial infections. The issue arises when we overuse antibiotics. So in the United States in particular, antibiotics are being loosely prescribed. One example is a colleague of mine, a couple months ago, got a tooth extraction. She wasn't infected, but they automatically prescribed her antibiotics in case she got infected. Right, so the antibiotics are being loosely prescribed, it's becoming an issue. I also noticed upon my arrival to El Salvador that you can buy antibiotics over the counter. And some issues arise when that is available because, number one, in order for it to work properly, you have to be able to self-diagnose yourself. So figure out if you have a viral versus bacterial infection. The other issue is you have to know what species of bacteria you're infected with because depending on what that is, depends what kind of antibiotic you get. So another use of antibiotics are with livestock. They're actually placed in the livestock's food to prevent infections. One reason is because their diet. So cows, for example, are fed corn as opposed to grass in the United States. And corn is an excellent source to grow bacteria, E. coli, which is why you hear of people getting E. coli poisoning from undercooked meat. So instead of changing the diet and giving them grass that would prevent the growth of E. coli, they decide to throw antibiotics in their food. So with the overuse of antibiotics, we began to see the issue of antibiotic-resistant bacteria, also known as ARB, that are increasing. So ARB are bacteria that are no longer affected by antibiotics. Now, as I mentioned previously, antibiotics are found naturally in the environment, which means that you'll naturally find ARB as well, as they've adapted to the environment. The issue arises when we overuse antibiotics and we're inputting them in extreme amounts into our environment, and that's leading to a dramatic rise of ARB. So over here I have a diagram of how we select for antibiotic-resistant bacteria. This diagram in particular is talking about an infection in the body, but the same concept holds true when you're also referring to antibiotic-resistant bacteria in the environment. So first, you have a high number of bacteria. The blue bacteria are the ones that are sensitive to the antibiotic, and the peach, pink, not sure what that color is, are the bacteria that are resistant to antibiotics. So moving over to number 2, this occurs once you input the antibiotic into that environment. The ones that were sensitive will die off, the ones that were resistant will remain. Now, jumping to number 3, you've created the ideal environment for these ARB to continue to multiply. And you also have the case of number 4, where bacteria actually exchange their DNA through plasmids, and they're able to give their DNA, their resistance genes, to other bacteria that initially weren't resistant to antibiotics. So this is an example of why it is highly recommended that you finish your full dosage of antibiotics. And the reason is you need to take your full dose of antibiotics to make sure that all the bacteria are gone from your system. Or else you can create conditions where you're literally incubating antibiotic-resistant bacteria inside of you. So ARB is on the rise, and this figure is specifically talking about Staphylococcus aureus, which is a species of bacteria. However, we've seen that it's on the rise of many different types of species. It's also comparing penicillin versus methicillin, which are 2 different types of antibiotics. and hospital acquired versus community acquired. But the main point of this figure that I'd like to point out is that it began really quickly after the introduction of antibiotics. So if you remember in my previous slides, penicillin was first introduced in the 1940s. Of course, you're going to have a more rapid rise of ARB in a hospital environment because you are concentrating people that are sick, that are taking antibiotics, but it's still on the rise in our community, which is what my talk focuses on. So here I have a video that shows exactly how quickly antibiotic-resistant bacteria are able to grow. And this video was created at Harvard University.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pours thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So the first time I saw that video, I was completely taken aback because that was in the time span of 12 days, and you saw that they were able to survive on 1,000 times the concentration of antibiotics. So with with rising antibiotic resistance, we see increasing impacts. The more obvious impacts are the public health challenges. If you have a decreased effectivity of antibiotics, then you'll have an increase of incurable infections. Some of the less obvious challenges are the ecosystem challenges. Antibiotic-resistant bacteria can disrupt bacterial signaling, which is the mechanism that bacteria use to communicate with their environment. It can also displace bacteria that are required for ecosystem function. So here's a diagram of how ARB spread. So initially, you have your antimicrobial usage. So let's say you take your antibiotic, maybe you get lazy and you don't finish your dosage, then you can harbor antibiotic-resistant bacteria. You can pass it along to other humans. The same thing goes with animals. They can pass it to each other. Humans and animals are able to pass certain types of bacteria that can infect both animals and humans. And then you have the case where if you eat undercooked meat that may have ARB, it can be transferred to humans. Then once you've eaten your food, then things happen and you have manure and also humans have sewage. And that can leach into your environment, which can spread ARB. In the case of Los Angeles, you could have sewage spills that can leach into the environment, but we also have an extensive homeless population They don't have access to restrooms, so all of their waste are entering the environment. So today, my focus is on the environment, the prevalence of it, and the system that I study. So one way that bacteria can spread in the environment are through watersheds, which is an area of land that drains into a lake, river, or other waterway. So to the left, we have an example of a watershed. As you can see, there are many different points in the stream that make its way down to a single point. And as it's traveling, it makes its way through different types of environments. So if you focus on the top left corner, we have a farmland. So remember, they use antibiotics in our livestock. And let's say there is a precipitation event, that water can bring the bacteria to travel through the watershed, ultimately making its way down. So if you have a natural, healthy watershed, the good news is that you can filter this runoff and have cleaner water. The issues arise are when you have an urbanized watershed. So here I'm comparing a more natural, healthy watershed to an urbanized one. So to the left, we have many ecosystem benefits with this system. First, it decreases water velocity, so it gives water a chance to seep back into the ground, and it recharges our groundwater. It also filters pollutants through sedimentation. Also, in the case of bacteria, UV light can be exposing into the water, which can actually decrease counts of bacteria. To the right, we have an example of an urbanized watershed. This is a concrete line system, and so what happens in that case is it increases water velocity. It doesn't allow for any water to infiltrate into the ground, so there's no groundwater recharge. And the main focus of my talk is that there's no more pollution filtration. So my studies took place at the Ballona watershed. It's 338 kilometers squared, and it's a highly urbanized portion of Los Angeles, and it's 83% developed to be exact. So what happens when you have a highly urbanized system is you have lots of pollutants. So pollutants from all around the area make its way down the watershed, ultimately ending up in the Ballona wetlands, which is also where I did my studies, and making its way to the Santa Monica Bay. So the Ballona wetlands are a coastal wetland impacted by urban runoff. It's one of the last remaining in Los Angeles County because it's been significantly reduced due to urbanization. Despite its degraded condition, it still offers ecosystem benefits. So over here outlines general ecosystem benefits that wetlands provide. We have instances such as bacteria that are able to break down contaminants. It allows for water storage. It also provides a critical wildlife for habitat for animals. But the main ones that I'm focusing on are that it filters contaminants and sediments, and it provides a cleaner water outflow. Another fact about the Ballona wetlands is that it is by the ocean, so it experiences tidal fluctuations. So what that means is that 2 times a day, it will experience a high tide, also known as a flood tide, and 2 times a day, it will experience a low tide, also known as an ebb tide. So a little bit about how this process works. We have water coming upstream from the watershed. So we have freshwater runoff. It mixes with the salt water in the Ballona estuary. During high tide, the water will get high enough to the point where the water can infiltrate the wetlands. There's a tidal gate that will close when the tidal height has gotten too high. So the water is actually sitting here throughout the day during sunlight hours. Then during the low tide, the water levels will decrease, and we have water flooding out, ebbing out, I'm sorry to be exact, back into the estuary and then out to the ocean. So this process has actually been studied by John Dorsey. You'll be hearing about that more in a moment. But what he found is that this process actually reduces bacterial pollutants. So this got us thinking of how it would affect antibiotic-resistant bacteria, to be more specific. So we came up with our study questions. The first is, what is the prevalence of antibiotic-resistant bacteria in the Lower Ballona watershed? So we wanted to see, first, what was out there. Second, there is a reduction is there a reduction in ARB when water is subjected to wetland processes? So we're going to be looking at the flood tide coming in, collect bacteria, see what the abundance of the resistance, and then we're going to compare it to water that's coming back out. So after it's been subjected to wetland processes, perhaps we will see a drop. That's what we were asking. The third is how would these levels of ARB compare with secondary effluent wastewater? So our reason for comparing it to this was sort of as a positive control. We expect to see high levels of ARB since we're dealing with treated human sewage. So we use that as a means of comparison. How do the environmental samples compare to the worst case scenario? Over here is our study site. We took runoff samples initially upstream. So this is straight runoff from our concrete line system. Then it will come down and enter the minor wetlands. Over here's a zoomed in version. So again, we take samples when the water comes in, wait a few hours, then we took samples of water that was coming back out during the ebb tide. We also did some initial studies at Darby Lagoon, and I'll show you a little bit of that data as well. So this is our methods in a nutshell. So we would take our water samples from the environment, bring them back to the laboratory, and we would do a heterotropic plate spread. So this allowed us to visualize the bacteria that were in our water samples. We also did a dilution series because we needed the right amount of bacteria. If we had too many bacteria, it would be hard to work with, hard to count. For example, this is a straight heterotropic plate spread with zero dilution. And you can see this plate on the right, it's too much to work with, so we would have to dilute it down, and then when we were doing our statistical analysis, we would bring it back up to what the actual numbers are. Another thing to notice is straight right off the bat, the comparison of flood versus ebb. So already we are seeing there is a reduction in bacteria. The only thing that this doesn't answer though is if they're resistant or not, which is what our question was. So our next step was to do a replica plating method, and we would take our original plate that we spread plated, and then we would subject them to all the antibiotics that we were testing, which I'll show you in a moment. But just a little background on how this works. So first we have a sterile velvet on a block, and we actually invert our plate and stick it onto the velvet so that the colonies from the master plate are transferred directly down to the velvet. Then we'll have a plate, a replica plate with no antibiotic, and then a series of plates that do contain antibiotics. So right after you inoculate initially, then you go in a series, you'll take your plates of all different antibiotics and introduce them to this velvet that now has bacteria. So in this example, you can see the one with no antibiotics had the 5 original colonies that were in it, but when it had a plate with ampicillin, only 3 of them were left. So that means that 3 were resistant to the antibiotic and were still able to grow in its presence. These were the 8 antibiotics that we tested. These antibiotics were chosen to represent a broad class of antibiotics. It also has some antibiotics that are derived from those that are naturally found in the environment versus ones that were synthetically created. So our next step was to take pictures of the results of our replica plates, and then we would upload them onto a computer and do a photo analysis of these resistant colonies. So we would use Photoshop and literally circle each colony. So over here I have an example of a plate that has ampicillin, which was assigned white circles. And then to the right I have a plate that has cefotaxime, which was assigned red circles. Then what we would do is take that image using our registration marks to make sure that the plate is aligned properly. We would overlap it onto a master plate, and then from there you could see, based on the color circles you have, how many resistances do each of these colonies have. Then we did a verification method. The one that I will go over is Kirby-Bauer, which is one that is a standardized method to measure microbial resistance. And here's an example of it. So in this method, you have a carpet of bacteria, and it's all from a single colony. Then what you do is you place antibiotics on them. So these are different kinds of antibiotics. And then if you see a halo, that means that they weren't resistant because the antibiotics were still killing them. But if there was no halo, then there was a resistance. So now we had all this data, and we had to figure out how to analyze it. So we first chose a cumulative frequency to compare environmental and wastewater samples. We used mean ranks to determine which antibiotic bacteria are most resistant to. And then finally, we did some species identification to identify what kind of bacteria are having these resistances. So first I'll go over cumulative frequency. So this is a really busy graph, and it's kind of hard to see exactly what's going on. But I'll explain pretty much what you should take out of this. So on our x-axis, we have the number of antibiotics resistant per colony. So since we tested 8 antibiotics, we have a number from 0 to 8. And then we have our cumulative frequency. So out of the 23 samples that we had, all kinds of samples, Hyperion and environmental, we were able to combine a lot of them using a statistical analysis. So if there was no difference between those samples, we were able to combine them. But if there were any differences, then we had to create a different distribution. For example, with our Eb1 sample, this represents 5 events, and our Eb2 only represented 1 event. So that's why we have these different lines. So the way you want to look at this is if you have curves that reach 1 more quickly or reach their asymptote very quickly, that means that that sample, those sample events had a low amount of bacterial resistances. If it reaches it really slowly, that means you have a water sample that had a high amount of resistances. So I'm going to clean this up a little bit and focus on the ones that we had the majority of events for, just so you can see the trend that we noticed. So what we can see is that, as expected, our Hyperion samples had the most amount of resistances. That was then followed by our runoff, so the water that was taken upstream, and also our flood samples. They were actually not statistically different. So it's showing that between the runoff to the water coming into the wetlands, there's no difference. But then once it's been sitting in the wetlands all day, being subjected to wetland processes, we see a drop in antibiotic resistance. So this is showing that wetlands are acting as a sink for these ARB. Next, we did a mean rank. And this was to determine which antibiotic bacteria are most resistant to. So the numbers, they feel a little backwards because if you have a rank of number 1, you will have the most resistances in your sample. And if you have a rank of number 8, you will have the least amount of resistances. So what we found is that ampicillin was the antibiotic that bacteria were most resistant to, whereas streptomycin was the antibiotic that they were least resistant to. Now, there still would have to be more studies to decide the cause of this, but it is a little interesting that ampicillin is one of the top prescribed antibiotics. It's been around for, I think, around 60 years, and it's used for more common infections such as urinary tract and respiratory infections. Whereas streptomycin is not as lightly prescribed, it's much stronger, and it's used to treat more intense, less often infections such as tuberculosis. So last, our species identification. We were able to identify the species of multiple bacteria, but to shorten it up for my talk, I'm only presenting bacteria that have been implemented in human illness. So one that's actually really interesting is Staphylococcus aureus. Depending on the strain of Staphylococcus aureus, it can be something more light to something really concerning. So I'm not sure if any of you have heard of flesh-eating bacteria. Well, Staphylococcus aureus is actually responsible for that depending on the strain. So if you have a bacteria that has resistances to antibiotics and it's eating your flesh away, it's going to be really bad. So the other thing I want to point out with this is we found the same species even from similar sources. However, they have different sets of resistances. So this goes to show that resistances seem to be more environmentally dependent as opposed to species dependent.